All right, welcome back, everybody. Today I'll talk about the FastQ, fastp, processing tool, which can do a great deal for pre-processing your raw sequencing files before moving on to an aligning tool, if that's the next step in your pipeline. If you guys find this video helpful, feel free to like and subscribe. It's beneficial to me, but also to other people who might be looking at using this tool. It'll help them out as well. So fastp appears, so I've never used fastp before, but some people that work in the same lab as me have used it before, and they find it extremely helpful. It's really useful for filtering out things like low quality reads, trimming, removing adapters, which is what the people that I know who use it for. It can split files, merge files, etc. Today I'll be doing kind of a quick little run through of it, as I found it pretty interesting and also really, like I mentioned, handy. I'll put a link to this page down in the description below. That way, if you guys want to run through kind of everything that I'm doing today, you can do that as well. So the first thing that we would have to do is install fastp. If we go on their page, there's actually a little install link here. So you can see it's in the Bioconda channel, and which makes it really easy for us, since we've been doing everything with miniconda, to install. And to install it, like it is on their page, you can do conda install -c, and then we need to specify that bioconda channel, and then fastp. And at the end, if we do -y, it'll automatically proceed once it reaches that step where you normally have to type yes or no, whether or not you want to install it. And since yes, we do, we can just do -y. And then it'll go through, do everything, and then install. The files that I'm going to be using today are from the RNA-Seq pipeline that we had run through before. So I'm just going to do 771 and 772. If you look there on the bottom, and I can't move my mouse, but right across the taskbar on the bottom, you can see that it's the link, and it's bioinformatics_YouTube/blob/master. And if we want to download them, we actually have to change that /blob to raw. And that's why I have this folder or this file here. So this has all the links to all those files. So if you go ahead and download or copy and paste this into a text document, so I pasted all of that into readlist.txt. You can see here, these are all the raw. What we can do is download them using wget. So wget has a really handy flag, which is I. And if we pass in that text file, it'll go through and download each one of them. So go ahead and run that. And I'll stop it after it gets the first two done, since we really don't need all of them. So we'll go ahead and kill it, and then we can clear this, come back, and we'll delete all of them except the 770. That's the one that we'll be practicing on today. All right. So now with all of that downloaded, we can actually start running and looking at fastp. So to get to the fastp help, kind of like any other tool, we can do fastp-h, and then we get all this information. And I'll kind of break down the different things that we'll use, and I'll compare it back to the termomatic that we had done before, but this time using the fastp way. For the input files of fastp, if you scroll to the top, you can see there are a couple different flags for input. If we want to do just single end reads, we would use this -i in the lowercase, -i and -o. So for single end, you only have to pass in the one direction. But if we want to do paired end, we have to do forward and reverse. So that's where we have to do the lowercase and uppercase. So if we want to do that, we would have to do fastp, okay? And then -lowercase I, and we would pass in that SRR 771, and then capital for the second read, or the reverse. And then for output, if we want to keep them in the same order, lowercase I goes to lowercase o. And we can just keep it nice and short, 1.fastq.gz. And then for 2, uppercase I, we need to pass in the uppercase O. Do the same thing, 770_2.fastq.gz. If we go ahead and run that, this is the most basic way of running fastp. And we get a bunch of our information here. We can see a bunch of reads past filter overall. No reads failed due to them being too short. There were some adapters, but overall looks pretty good. Okay. By default, fastp uses the length of 15 for checking these adapters. No, for removing reads, I'm sorry. And if we want to increase that, we need to set the -L flag. So if we go back up here, we can increase -L to like we had before, which was 36. So now any read, after all the processing and stuff, that is less than 36 base pairs, it's not going to keep. So if we go ahead and run this, we can compare that reads too short here. So above, remember we had 0 reads that were too short, and now we have 120. So 120 were between 15 and 36 base pairs. Pretty straightforward. Something that is also useful from fastp is adapter trimming, which happens automatically in fastp with, I think it's just forward end or single end reads. It's really nice compared to with Trim-O-Matic, where you have to specify where the adapter file is, and then kind of all the different things that we want to go along with the adapter. So it's way more handy with fastp. Usually there's no harm in running the adapter trimming whether or not there are any adapters identified in a program like FastQC, so leaving it on here isn't too big of a deal. Like I had mentioned, it appears that it only runs with a single end read automatically, detecting the adapters in the first million reads or so. It does not auto-detect with pair end reads by default. If you would prefer to specify like where your adapter file is, so here I have like an example adapter file from their page. Let me see if it opens up here. You can specify that using the flag, what was it? Adapter fasta. And then you would feed in that adapter fasta file that has those adapters. We'll back out of there. Additionally, you can feed in, so here we have Illumina, we have our forward adapter and our reverse adapter. You can feed those in if you want independently. So here we can do adapter sequence, and then we would be able to type in this. I think it needs an equals. And then if we want to do adapter for the second one, we have to do adapter _sequence, sequence _read2, and then equals whatever that reverse is. So this is if you're going to specify what your adapter sequences are. Like I mentioned, it's not automatically on for paired end reads, but when it does do it for paired end reads, if you want it to automatically identify the adapters, it does what's called overlap analysis. So if there's some overlap, then it'll remove those that it determines are adapters. And if you want to do it for paired end, I believe the flag to do that is detect adapter for PE. And it can take, okay, there you go. So no adapters were detected for paired end. So go back. We'll keep working with this same line for kind of everything that we go through here. So there are some other options with FastP for trimming, like sliding window trimming with the kind of like what we did with Trim-O-Matic for removing the beginning or end bases that are poor quality. These flags are cut, I don't know why it does that, cut, and then for the forward end, front, and then if you wanted to do the tail, it would be cut tail. But then there's, for these, you have to specify, you don't have to, there are defaults, but might be good to specify cut forward window size. So cut forward window size, and that defaults to 4, and here you could do 4. And then also cut, it's not forward, front. Cut front mean quality. So of that sliding window, what's the mean quality that you want? And the default is 20. And like we were talking about for DNA sequencing with the whole genome, we would probably want something like 25 or even 27 to keep that up there. So this is cut front. You could also do cut tail window size and quality, and they default to the same thing, 4 and 20. There's also a flag that's called cut right. So if we back out here, and what this is, it's similar to the sliding window option for Trim-O-Matic. So starting at the beginning, what is the window quality that you want and what length of window do you want? And this also defaults to 4 and 20 as before. If you enable cut right and cut tail, then cut tail. So remember, cut right and cut tail. So cut right is going to be that sliding window, and cut tail is going to be cutting from the end. If you enable those both, the cut tail isn't really going to be of much help because cut right is going to start on the left-hand side and do a sliding window across. And as soon as that quality drops, it's just going to get rid of the rest of the read. So their cut tail isn't really going to be used. Additionally, if you run cut front and cut right at the same time, so cut front, it's going to run cut front first. So it's going to remove the low quality reads on the front of the read before moving on to cut right. If you remember with Trim-O-Matic, the sliding window, and like I mentioned not too long ago, the sliding window of cut right, once the quality is below whatever that threshold is, by default it's 20, it's just going to get rid of the rest of the read. Well, if the first few base pairs have a really poor quality and you use cut right, you're going to get rid of basically your entire read. So at first it's going to do the cut front. So it's going to remove however many base pairs are poor quality at the front of the read, and then cut right will come in and finish off the rest of the read. The problem, if we run this now, we will have very few reads that are kept. Because we have that, yeah, well, it's not too bad actually. Because we're using cut right. So the length drops below 36 for a lot more reads now. We can see here 150,000 reads drop below that 36 base pair threshold that we have set. We can go ahead and keep that and keep working with this line now that we're satisfied with it. Next, FastP can do what's called base correction using an overlap analysis. So of the first forward and paired end read, you can have some overlap with your read, right? For base correction, you have to have this overlap with paired ends, which usually requires longer read lengths with a short insert size. So if your insert, so think of it as you have your read length. So right now we have, say, a 50 base pair read length. If our actual insert size is much greater than that, say 300 base pairs, and we're only taking 50 base pairs from each end, there's not going to be any overlap there, right? So this usually works, I would say, better with longer reads because you're going to have more overlap. If there is, so going back to this read correction, if there is overlap and there's a disagreement between between a base pair between the two reads, then FastP will do a base correction from the low quality read to the high quality read. So if one of the reads has a really bad quality and it's different than the one with really high quality, it'll flip the bad quality to high quality. This requires a few things though. For base correction, there needs to be a fairly high overlap between the two different reads, and it has to be paired end. And then there's a limit, but there's a max amount of disagreed bases that are allowed between the two reads. And by default, that's 5 base pairs between the two reads for not matching. And then 20% is also another flag that you have to see. So if we want to do this, the base correction, you can use -C for that. We go ahead and run this. So we'll do all the trimming, and then now we have 51 base pairs in 51 reads that were corrected from this overlap, which is pretty cool. Next is poly-G trimming, which is really important for certain sequencers, more specifically for 2-color sequencers. So with 2-color sequencers, you have 4 combinations of colors, right? So you can have, of the two, one on, the other one on, both on, or both off. Well, for NextSeq and NovaSeq systems, this no color is actually a G base pair. So if there's no base that's being read, it's still going to be recognized as a G, which may or may not be there. And so it could be a good idea to just remove any of those that are at the end of a read and repeating Gs. So no signal at the end of a read. This is automatically enabled for those systems of NovaSeq and NextSeq based on the machine identifier in the read file. But if you want to turn it on, it probably isn't going to hurt too much. And to do that, you can do a -G. And if you have NextSeq or NovaSeq reads and you don't want it on, it would be a capital G. So on is lowercase, off is uppercase. But here, we're just going to keep that out. Next, FastP has the ability to process UMI on reads, so unique molecular identifier on reads, but I've never done any UMI index reads. From my understanding, each read has a unique identifier, which then allows you to trace it back to its origin or reads from the same molecule. To enable UMI processing of or in FastP, you can use the flag -U -U for UMI. And then when you do this, you have to specify the location, which can either be read 1, read 2, if it's paired end, and a couple other ones, which can be found on their page. And we'll scroll down to that. UMI. So it could be index 1, index 2, read 1, read 2, so on and so forth. But we don't have that here. But then you have to also specify the length. So if you do -U, you need to have UMI_LOC, which would then be one of those locations. And then you also need the UMI length, and you have to tell it how long that unique molecular identifier actually is. Since we don't have that for ours, we're going to keep that out. And then something else that's kind of handy here for FastP is overrepresented sequences. So back when we did our FastQC video, I believe I talked about overrepresented sequences and when they may or may not be expected, and when they could or could not be a big problem. In DNA sequencing, you probably wouldn't expect many, if any, overrepresented sequences unless you did some kind of enrichment like ChIP-seq, because then you're selecting out a subset of reads from basically the whole genome. So for DNA, you probably don't want those overrepresented sequences, but for RNA, it's kind of expected that you're going to have overrepresented sequences, especially if you're going to do something like an expression analysis, because if you have reads that are genes that are upregulated, you're going to expect to have more reads from them than genes that are downregulated. So to get this overrepresented sequences, you can do fast and then lowercase p. And then what it's going to do is it's going to sample the reads in your file to look for overrepresented sequences. And the default is 1 out of 20 reads. So you can change that by doing capital P and then setting that equal to whatever you want. So if you wanted to look through every single read, you would do 1, since it's 1 out of whatever number you put in. If you have a really big read file, you can do 1 out of 100, and it would cut down on the time that it takes to process all of that through. But since here we have RNA sequencing files and I'm going to expect overrepresented sequences, I'm just going to leave that out. And finally, so we did C for error correction, yeah. So finally, the last thing that I'm just going to mention here quick is going to be the case of interleaving forward and reverse sequences. I don't know off the top of my head where this would be used, but I'm sure there are tools out there that would use the interleaved forward reverse or paired end reads. If you want to use fastp and then have this be the output, you can use your output kind of here and then do merged out and then have, I don't know, 770_m.fastq.gz. And the merged reads will go here. The reason why you need to have this -o, and then you should probably also have unpaired and unpaired 1 and unpaired 2. Unpaired 1 and then 770_1u.fastq.gz. Oops. And then unpaired 2 2u.fastq.gz is because if there is a problem when trying to merge the forward and reverse into this one file, then it'll spit it right out into the unpaired file. And then for unpaired, or not the unpaired, the single direction file. And then for unpaired, say you have forward reads that fail reverse or reverse reads that had a forward fail, it'll keep them in this unpaired file. So if you do something like top hat, for top hat you can input forward paired, forward reverse paired, and then forward unpaired and reverse unpaired. So I haven't run this, but let's give it a shot and see if it works. Oh. So we need to enable -m, which makes sense. So we'll go before merge out and do -m. Okay. There we go. So now we have our forward unpaired reads and our reverse unpaired reads that had the other one pass. So I think in the future I'm going to be using fastp just because it seems like it combines FastQC and Trim-O-Matic kind of into one tool, and which should save some time, and also it's just way easier for creating a pipeline with that. But so yeah, if you guys found this helpful, feel free to like, subscribe. If you have questions, maybe I didn't cover everything or cover something as good as I could have, leave them below and we can have a discussion and maybe better ways of going about processing with fastp. So thanks for watching, guys.